My name is Claire Barlar, and I will be presenting on the article Leveraging an Open Science Drug Discovery Model to Develop CNS-Penetrant ALK2 Inhibitor for the treatment of Diffuse Intrinsic Pontine Glioma for Organic Chemistry at Wheaton College. A brief overview of what this presentation will look like. We'll start with an introduction, what the biological problem is, the hypothesis of the researchers, synthesis for the organic chemistry, results of the experiment, and the conclusion. A brief introduction. So there is currently no strong treatment for diffuse intrinsic pontine glioma. I will explain what this is, but very briefly, this is a very aggressive cancer in pediatric patients. So we need to develop a CNS-penetrant ALK2 inhibitor. ALK2 is an activin receptor-like kinase. It is a protein, also a type 1 receptor for bone morphogenetic proteins and other members of the transforming growth factor beta family. So this is actually a mutation within the gene that led to DIPG in the brain. So we need to do some modifications. We need to create a compound that is potent enough to attack the ALK2 protein and selective enough so it doesn't hit any other proteins in the brain. It also needs to be permeable in the blood-brain barrier. So there are actually side effects if it attacks other proteins, like I said. So what we want to look for in a compound that we make, it has to go through the concept of ADME, which is absorption, distribution, metabolism, and excretion in the brain, and also PK, which is a pharmacokinetic. So is it bioavailable and is it attainable for people who are diagnosed with DIPG? It's critical for the CNS drugs to contain these because it dictates what the drug's ability is to actually cross the blood-brain barrier to reach its target and exert its effect while minimizing any other side effects. The biological problem is DIPG. This is diffuse intrinsic pontine glioma. Like I said, it's a very aggressive pediatric cancer found in the pons region of the brainstem. It makes it almost impossible for any cancer treatment that we know of, such as chemotherapy, because chemo cannot cross the blood-brain barrier. And around 200 to 400 children each year are diagnosed with this, and usually it's ages 5 to 9. It is almost uniformly fatal within 9 to 15 months of the diagnosis, and only 10% survive 2 years after being diagnosed. Although this type of cancer is very rare, it's actually one of the leading deaths among children in cancer. So we need this research to prevent more innocent children from losing their lives. As a future doctor myself, who is very interested in pediatrics, I knew I needed to study this because it's a growing problem. There's no treatment for this, and I don't want to see any more children who don't understand the concept of cancer to die from this. So current treatments right now for these children is radiation. It can shrink the tumor and mediate some symptoms, but it is not an effective treatment and it won't prolong their life. Some steroids are also used to control symptoms and swelling of the brain. And then emerging treatments such as this one, a targeted therapy to attack the actual ALK2 proteins and reach the pons, as well as immunotherapy and ultrasound is being tested and looked into. Some current inhibitors, these are not necessarily relevant for what we will be exploring today, but it just gives a brief overview of what compounds are being used to attack these proteins and why they're not working. So let's look at what will work. Or the hypothesis of this study is that they need to develop a series of potent, selective, orally bioavailable, brain-penetrant ALK2 inhibitors to produce preclinical compounds capable of treating diffuse intrinsic pontine glioma. So the orally bioavailable part is extremely important in this because we cannot develop something that will work extremely well but be very unattainable for researchers, doctors, and especially the patients to get their hands on. So how has DIPG been treated? Like I said, there's been previous inhibitors such as LDN-214117 and LDN-213884. I will explain these more in depth shortly, but they have a low toxicity and have an increased selectivity to proteins in the brain. Other ways that DIPG is tried to be attacked from is attacking other proteins in the brain, such as ALK1, 3, and 5. But they do come with side effects if they are attacked. as well as changing previous molecular weights could help with DIPG, or where the hydrogen bonds, and what accepts and what donates. The first synthesis is of LDN-214117. This is a pre-existing inhibitor of the ALK2 protein. It's genome-wide selective, so it can be highly specific on what target it hits, as I previously mentioned. It has a low toxicity, and in a study, it actually showed a 15-day prolongation in rats. It had an increase in potency and an increase in selectability. The next one was LDN-213844. This is also known as an ALK2 inhibitor. It's also very potent. It reacts strongly with the hinge region of the structure at the C4 carbon, and it's actually favorable bond interactions. So there's a picture of LDN-213844 on the right. Our first synthesis will be forming compounds 1A through 6E. So you can see on these photos, there's examples like X1, X2, and R1. And if you look below these compounds, you can see what substituent is being placed there. So for example, 1A, at R1 you have a methyl group being attached, X1 you have a bromine, and X2 you can add a chlorine. And all these have been tested against, but in this specific compound, we want to turn our eyes to compound 6A, which adds a methyl group to R1, and then a fluorine to R1 as well, and that was tested. So this is actually a Suzuki coupling process, which is a palladium-catalyzed carbon-carbon bond-forming reaction between an organoboron and organic halide, which forms a new carbon bond. So in part A, we have a boronic acid, which is a coupling nucleophilic partner, and then a catalyst and a base to activate the boronic acid and a polar solvent in heat. In process B, it uses an aryl boronate ester instead of the free boronic acid, but it still has the same concepts in a Suzuki coupling: a nucleophilic partner, catalyst, and a base, and a solvent in heat. In process C, the tert butoxycarbonyl, or BOC, is removed by the 4 molar HCl at room temperature for 30 minutes. And this will actually give the product HCl to the amine. The second synthesis we're going to look at starts with the same compound at 1A and uses similar intermediates, but we're going to end at 11C. And one compound you want to turn your eyes to is 11B. In this process, it uses Suzuki-Moore cross-coupling as well. Like I said, it starts with a nucleophilic partner and then a pre-catalyst, and then contains a base reaction by sodium to activate the boronate for transmetallation, and then uses DMF in water at 100 degrees Celsius as the solvent, which this will form an sp2-sp2 carbon bond between the aryl B-pine and aryl halide electrophile. In step B, it is a repeat of the Suzuki coupling. So you have the nucleophilic partner, catalyst, base, but this time is done by potassium and a strong solvent. Then in step C, we have 4 molar of HCl, which initiates the acidic BOC deprotection and becomes an amine and the HCl salt. In the last synthesis, we have the same compounds, and this time it actually uses same intermediates, but this time we're going to go all the way to 14G. Some compounds you want to turn your eyes to is 14B, 14P, K through I, and G. In this synthesis, we will develop the CNS-Penetrant ALK2 inhibitor. We will use previous intermediates, like I said, such as 3A and 6A, to incorporate the C3 position in the C5. As well, we will continue to use Suzuki coupling, which now, hopefully we know, is the boronic acid as the nucleophilic partner, the catalyst, a strong base for transmetallation, and a strong mixed solvent in heat. Moving in step B, we continue with Suzuki coupling with the same process. Step C also follows the same reaction in that the BOC group is cleaved off and replaced. Although in this synthesis, you may have realized that we have more steps. In step D, we have a nitrogen alylation by an SN2 reaction, which we studied in class, and it actually uses tosylate as the leaving group for the electrophile. We know this to be a very strong leaving group. Then the triethylamine acts as a base to neutralize the proton produced when it was alkylated. The C-nitrile and heat are conditions that are used to promote the SN2 displacement reaction when the nucleophile is not necessarily ideal. Lastly, step E, this is a sulfonation step because the nucleophile is an amine and the reaction will give a sulfonamide to keep it nucleophilic at room temperature. Some results. After synthesis took place, they measured the reactivity with selection potency and permeability for 6 compounds that gave positive results, such as 6A, 11B, 14G, 14K and I, and 14P. Rats were tested with these compounds using in vitro PPB, which tests in the body of an animal for plasma and protein binding. This was done in rats, like I said, but also in human. Each compound was relatively stable to an extent, and from the data presented in vitro, it was found that PPB cannot actually determine the concentration of the plasma. So we had to move to a different study, in vivo PK and ADME. This was a testing where there were no adverse side effects amongst female mice, although compound 14G registered with a half-life and a bioavailability of 7%, which is not reliable or what we want to look for in a compound. This made researchers drop 14G from their study. It was also found in vivo by adding HCl to compound 6A and 11B that there were more improvements than the free base did. And by ADME testing, it was found that 6A, 11B, and 14P were most bioavailable than the LDN 214117, making them extremely well-liked to continue with future testing. Through biochemical assays, researchers were able to find what compounds actually inhibited the ALK2 protein and what activated it. This led them to begin processing which of the 4 remaining compounds would be able to pass through the blood-brain barrier. Through their tests and trials, they found that compounds 6A, 11B, and 14K were more bioavailable, potent, and selective, and had a high brain-to-plasma ratio that could pass on for further research, comparing current targeted therapies to see any movement towards finding a reliable treatment for diffuse intrinsic pontine glioma. So here you see that we got rid of 14I and 14P, and we moved on with 6A, 11B, and 14K. We have now learned that LDN 214117 was expanded around the 3,5-diphenylpyridine inhibitor class in order to optimize the potency against ALK2, but retain selectivity. That way, LK5 is maintained. From that, 6A actually showed a greater potency with the methyl on the C4 position and still avoided the ALK5 protein, which would have given side effects if it was attacked. And even off other off targets with minimal activity with the H-ERG potassium channel in the brain. At the C3 and C4 positions, it yielded 11B and 14K, which both similar to 6A had favorable characteristics. As well, it had a very high blood-brain barrier penetration, which is vital. Now knowing that these compounds contain vital characteristics for blood-brain barrier and selectivity, further research can be done with the current ALK2 inhibitors, proving the original hypothesis of the scientists. Thank you so much to listening to my presentation about diffuse intrinsic pontine glioma and what compounds are being formed. That way new research can come about and hopefully some cure and treatments can be used for the pediatric patients. I hope you learned something today. Thank you so much.